What I didn't say before is that the tip is very small, you know. And if you make a small tip, you need also a small spring. But it's quite difficult to manufacture small springs. So instead of using springs, they decided to manufacture cantilevers. Small cantilevers, if you deform them, they apply a force exactly like a spring. So all this system is microfabricated. So now once we have this, our cantilever, our tip, we need also a way to detect the deformations of our spring, isn't it? So how to make this detection? There are different ways. The one which seems to be the more efficient and the cheapest one consists in using a laser diode that illuminates the very end of the cantilever. So if the cantilever is deformed, the laser beam will be reflected at another place, okay? So then we have this photodiode here, which is composed of two segments. And depending on which segment is more illuminated, we can calculate the deformation of the cantilever. So here is an image that, actually a cartoon that draws this technique. Here you see you have the cantilever. This is our tip. It is illuminated by this laser. And the photodiode is here. This photodiode has two segments, a segment A and a segment B. If we are in between, you see A and B are equal. If we subtract them, we obtain 0, and it's what we see here. If we deform the cantilever upwards, in this case, the A photodiode will be more illuminated, and we know that our cantilever is deformed upwards. If we deform it downwards, in this case, the B photodiode gets illuminated more, and we know that we are deforming in this direction, okay? So this is the very simple system that permits to know the deflections of the cantilever. What is interesting in all this is the extreme sensitivity. You know, we can detect oscillations, deformation of the cantilever, which are below 0.1 angstrom. So a carbon-carbon bond is 2 angstroms. We go 0.1 angstroms. So it's extremely sensitive. So the atomic force microscope is called a microscope. Why? Because you can make images with it. And how do we make images with such a sensor? It's relatively simple. We just make a scanning of all the surface of our sample, and you see we are looking at the deformations of the cantilever. Okay, we have the laser system with the photodetectors that are here. So for every single point here along the scanning path, we look at the deformation of the cantilever, and this way we can reconstruct the surface topography of our sample. So a couple of words about the tip and the cantilever. So typically the tips are very small. Cantilevers are a bit larger. Here you see about 200 microns, but nowadays some cantilevers are short as 3 microns, because if you want to make high-speed AFM imaging, in this case you need very small cantilevers, and so as you can see, the range can vary between 3 microns, in some extreme cases up to 200 micrometers. So what about the tip? The tip is here. It is this small pyramid that you can see at the very end of the cantilever. The base of this pyramid is 5 micrometers. So it's a bit smaller than a red blood cell. And the radius of curvature, it is quite small. It's about 10 or less nanometers. Okay? A bicycle wheel is about 30 or 40 centimeters. This one is 10 nanometers. Since the tip is scanning the surface, the shape of your tip will determine the resolution you have. The sharper your tip is, and the better resolution you will obtain. So there is some kind of race to obtain the sharpest possible tip. So here are different possibilities. You can make your tip sharper. So here we are looking at the very end of the pyramid, and you see some guys are growing tips atop the tip. You see? So for doing this, it's relatively easy. You just need an electron microscope, which is quite old, the dirty one. And then you put your tip in there, and you focus the electron beam onto the very end of your cantilever. And all the dirt that is in the microscope will be focused and will deposit onto the tip, and you can obtain such nice ultra-sharp tips. You know? So this is one of the methods. Another method, which is more sophisticated, consists in attaching nanotubes, in gluing, sticking nanotubes onto the very end of the tip, and then you obtain very sharp tips. So we can have very sharp tips, but we need also to move our sample or our tip atop of the sample to have a good resolution, isn't it? It's not enough to have just the tip, but we need also very precise positioning system. So we could think about using motors, okay? But motors are not precise enough. There is another possibility that has been adopted quite early in the life of the AFM or the scanning probe microscope. These are these piezo crystals. So piezo crystals are very practical. Why? Because if you put them under a given potential difference, you apply, let's say, 5 or 6 volts onto the crystal is going to deform, you know. And so since we can control very precisely voltages, we can, with the help of these crystals, control very precisely also the movements. And just to give you an example, in usual AFMs, to have a motion of 100 microns, 0.1 millimeter, you need to apply 400 volts. You know, so if you apply just a couple of millivolts, you know, you will have displacement, very, very, very small displacement. So these piezo crystals made some kind of conversion, you know, of voltage in displacement, but you can also use it as in the opposite. You put a pressure on them and they generate some potential difference. It is what is used in these lighters, okay? When you turn it, you apply a pressure on it, it generates a huge voltage, and you have, you can light the gas. So this is this technique that is used to make the sample moving underneath the tip. So here is how it is implemented. You see here we have our piezo crystal. If we apply a voltage, the crystal will deform. In order to make displacements in the 3 axes, you can stick 3 different piezos, one in each axis, and then you can control the position of your tip very easily. Another option is to use cylinders that are piezo cylinders and to have electrodes deposited on its surface like this. And by applying the correct voltages, you can obtain movements in every axis. So this is the simplest and the most used option nowadays, to move the cantilever or to move the sample. Here you can see one of these piezo. These piezo are relatively expensive. The problem is that they are quite sensitive to liquids. So if you are working, you know, with biological samples, sometimes you have liquid that goes in there. If you have 400 volts, you can imagine it costs you about $20,000, you know, if you burn the piezo. So how do we make images with these devices? It's relatively simple. You see, you have the cantilever that is scanning the surface, and at the same time we record the deformation of the cantilever. So we are scanning here. Once we encounter a small mountain, you see the computer recognize this and it makes you a small line. Then if you go into a small valley, it's recognized, and we build a complete image of the surface of the sample. So here is an image of a typical AFM. It's an old one. At the time it was costing, or also nowadays it costs you about $200,000. You see, What is relatively cheap? If you compare to an electron microscope, you have for 1 or 2 millions, you know. And I have friends in Italy, they build for $50 small AFMs that have quite high resolution. So the instrument is relatively cheap. In addition, it's quite small. Okay, why is it small? It's because we don't want this instrument to be shaken by the building or by structures all around. So if you make it smaller, it will have a resonant frequency that will be very high, and in this case you will be safe of the motion of the building and the structures that are all around. You see, so it's interesting to make them as small as possible. So as I said, the instrument is relatively cheap. As you see, the working principle is relatively basic, but even with this, you see, you can make astonishing images. You can see here the organic compound, and here is the image with AFM that you obtain with it. So you can see single molecules. I mean, it's quite incredible for such a simple instrument. It is one of the possibilities that you have with this microscope. You can make very high resolution images in vacuum, in air, or in liquids. So that's something very interesting. The instrument in the beginning of the 90s was found to be also quite useful for measuring interaction forces between single proteins. So it's quite complicated, isn't it? We were talking about imaging, and suddenly, okay, we go to measure interaction forces. How is it done? It's relatively simple again. You see, you have your cantilever, and then at the very end of the tip, here you attach one protein. And on the mica, on the substrate, you can attach another one, another protein that is attaching to this one, okay? They like each other. So you go down, and when you are close enough, the two proteins bind each to other, okay? And then when you move backwards, after a while, okay, when you extend the two proteins, the cantilever will start bending downwards. Why? Because the two proteins are holding it attached to the substrate. But once the retraction force of the cantilever overcomes the attachment force between the two proteins, the bond will break, okay, and the cantilever comes back to its rest position. So by using this simple phenomenon, we can calculate the interaction force. So how is it done? Here we repeat the same process. We have the cantilever, it goes down, the two protein attaches here, you see, then we go back, and when we are coming back here, the cantilever starts to bend downwards, okay? And when the bond break, in that case, the cantilever comes back to its rest position. The graph that is represented in red is just the deformation of the cantilever as a function of the distance at the height, you know, from the base of the cantilever to the substrate. So when you have a unbinding event, you have a very specific signal that appears here, and the altitude, the length of this signal here in the y-axis gives you directly the interaction force between the two proteins. So we have a very simple way also to measure interaction forces between single proteins, one single protein, and you can know how it is attaching strongly to any other protein you are interested in. So this is one of the possibilities you have to use the instrument. The third one is this. You can also have an idea about the stiffness of your sample. You know, when you go to a medical doctor, he will also very often push on your abdomen, okay, to see if there is something hard hidden underneath. So we can use the AFM tip in the same way. We can push onto the sample and look if the sample deforms or not. Okay? If the sample deforms under the pressure of the tip, we know it's soft. If it doesn't, we know it's very hard. So the experiment is done in this way. We have our sample here, and we look at the deformation of the cantilever. So let's say you have something very hard, okay? You push 1 millimeter downwards, okay? And your cantilever will also move 1 millimeter upwards. Why? Because it cannot penetrate into the sample. So you will have a slope of 45 degrees here. And you know, okay, if I see such a slope in these curves, it's something very hard. But if our sample is soft, the tip will penetrate in there. And this slope will not remain like it was, but will become something like this, you see. And by looking at the shape of this curve, we can say, okay, we have a hard substrate or we have something soft. It's, as I'm explaining, it's very simple, I hope. But behind, you know, the problem is quite complicated because you have the shape of the tip that you need to know. Then your sample has to be very homogeneous in order to have the calculation made properly. So it can be quite a complex problem. But the principle is this, okay? So here is an example of such an experiment where you can see here this force curve here. This should be at 45 degrees of inclination, okay? But with the units, it looks like a more or less vertical. The deformation is this. This was recorded on glass. But if we go onto a mitochondria that you can see here, in this case you see this curve that was almost vertical here becomes so flat as it is here. Okay? So we can say, we can measure the stiffness of our sample relatively easily. And here is an image that shows you the combination of all these three possibilities of the microscope. Here we are looking at an axon. So it's a nerve cell. The axon has a diameter of about 1 micrometer. Since we are using the EFM, we have the topology. Okay, we know at which altitude it is situated. The 4 colors are presenting the stiffness. What is red is hard, what is blue is soft, okay? And in addition, we attached a protein onto the tip, and when this protein interacted with some proteins present into the membrane of this cell, we have these events that happened, you know, and we could deposit these arrows there. So we know where some specific proteins are located, we know the stiffness of our sample, and we know its topography too. So that is the use of this microscope is mostly nowadays used for imaging, measuring interaction forces, or measuring the mechanical properties. A couple of years ago, we found that we can also use it for a completely different task. It is detection of nanomotion. This project was started because we got funding to find a way to detect bacteria that are resistant to antibiotics. And so we started to different technologies. We tried to look at different possibilities we have to detect this type of bacteria. Hopefully we were relatively successful, so we started quite a new domain of research in our laboratory concerning this nanomotion detection. So why do we think that it can be important is maybe explained by this slide. So if we are here all together, it's thanks, of course, who organized this school, but not only. We shouldn't forget Edouard Jenner, Louis Pasteur, and also Fleming, because, you know, if we can live longer nowadays, it's thanks to the discovery of vaccination, the discovery of hygiene, and the presence of antibiotics. Only these three discoveries are responsible for 80% of our surviving rate. You know, that is much higher than it was before. So our life expectancy nowadays is essentially due to hygiene, vaccination, and antibiotics. Unfortunately, antibiotics are becoming more and less efficient. and it becomes really a problem. Very probably it didn't escape your attention. Very recently in Pennsylvania, they found bacteria that are resistant almost to every antibiotic we have. And so this explains why Swiss government, at least, all the governments too, start to invest money, in this to address this problem. So how can AFM help in this topic? So if we look at the development of resistance to antibiotic, there are several causes, okay? There are different reasons. The first one is evolutive pressure. If you have antibiotics in nature, only bacteria that are resistant to this antibiotic will survive, okay? We can't do nothing about this. There is also another problem that is more political. It's that the pharma is not interested into developing new antibiotics. And why? Because antibiotics are so efficient. You give just for 1 week antibiotic to the patient that he is cured. It's finished. You don't need to give it. And so there is no money coming in, So they don't invest too much money in there. Also, if you go, for example, in Indonesia or in Malaysia, you can buy antibiotics into drug stores. You go into the department store and you can buy your antibiotics. And it favors the development of resistant strains. These three domains are nothing, we can do nothing about that, But there is another one in which we can change something, is the overuse of large spectrum antibiotics by medical doctors. So why do medical doctors use large spectrum antibiotics that develop this type of resistance? So it's explained on this image here. When you have a patient that arrives into the hospital, according to the symptoms of the patient, you can't say, okay, it's an infection, but you don't know what is the germ that is responsible, and you don't know how to treat him. But you cannot let the patient without treatment. So what you will do is to take some samples that you will cultivate, okay, and you will try to grow these samples in presence of different antibiotics, and then you will see which one is resistant, which one, okay, is more efficient to cure the patient. But to have the results of this antibiogram or this resistance test, you need 24 hours in the best case, but it can last for 1 month with tuberculosis or bordetella. Very long time, and during all this time, you don't know which is the best drug for the patient. And you need to give these large spectrum antibiotics that make resistance. So our aim when we started this project was to reduce this time, during which this large spectrum antibiotic is given, and to have a test that gives you a response much faster. And this test is based on atomic force microscopy. So to illustrate you how it works, it is a very simple game that you can try to do this summer if you go to the swimming pool. You go on this diving board, if you stick a small mirror underneath and then you illuminate the mirror with the laser, you will see that the laser beam is deflected if anyone comes onto the diving board, and any slight movement, any movement of this person will induce huge oscillations in this laser beam that is reflected. So this is the technique that we actually use. Instead of having this lady onto the cantilever, we have, of course, our nasty bacteria on it, and we are looking at the motion, at the shaking of this laser beam. It should be noticed that actually this system is not very new. The Japanese already invented something similar, and if you visited the castle of the shogun in Kyoto, in Tokugawa shoguns, they had an alarm system that was based on this principle. They made the floor of this castle in a way that you cannot walk on it without making noise. And the slower you walk and the more noise you make, And so it was a very efficient and very energetically interesting alarm system, you know, and we are using something very similar with our AFM. So a typical experiment looks like this. First we take a cantilever and we immerse it into PBS, into a serine phosphate buffer. And if there is no bacteria on the cantilever, here you can see that the cantilever doesn't shake, doesn't move. Okay, here we have the time, and the deflection of the cantilever in nanometers is here. There is a small slope, it's due to the thermal drift, but nothing serious. But then if we inject bacteria, and if this bacteria gets stuck onto the surface of the cantilever, we notice that the cantilever starts to shake. And later on, if we inject an antibiotic that kills the bacteria, we see that the motion almost stops completely. Okay? And what is interesting is the time frame in such an experiment. So I come back again. Here we have the motion of the cantilever in PBS with no bacteria. Then we move with bacteria, okay, onto the cantilever. Here we give some food to the bacteria. They are very happy. They move even more. You don't see it here, but you can see it on the histogram, which is the variance of the signal. The higher it is, the more the cantilever shakes. And then once we inject ampicillin, we see that the motion stops. And if we give food again, okay, nothing happens. So it means that we can distinguish, you see, the motion in 5 minutes. In 5 minutes, it has changed from this up to this, and it's very quick. It's really something quite interesting. So what is going on if we use, for example, bacteria that are resistant to this antibiotic? So we took exactly the very same bacteria, we mutated it to be resistant to ampicillin, and we made this experiment again. And here you can see again here the cantilever without bacteria, the cantilever in PBS with bacteria on its surface. It's shaking. It shakes even more if we add the food. But once we inject the antibiotic, the motion drops. You can see it here, but recovers after about 20 minutes. So we see the bacteria literally adapting to this new antibiotic and developing very probably all the machinery they need to resist to the antibiotic. So here the very first experiments were made with E. coli. So E. coli has flagella. And if you look at E. coli under an optical microscope, you can see them swimming left and right. So it's logical, okay? You stick this swimming bacteria onto your cantilever. If they shake, the cantilever will shake too. But we made the experiment with Staphylococcus aureus, which is a non-motile bacteria. It shouldn't move. If we stick it onto the cantilever, you see the cantilever, if the bacteria is alive, it will induce oscillations. But if as soon we inject an antibiotic in there, to which the bacteria is sensitive, the oscillation stops. And so we have, we think, a method that is quite efficient to detect sensitivity of bacteria to different antibiotics. We made double-blind experiments with the Hospital of Lausanne. So they gave us bacteria. Yes?
Can I ask you to show past and past?
This one?
And 43. If you add ampicillin, they're going to die.
Yes.
And in wash 53, it means that bacteria died.
They are still on the cantilever.
But died.
Dead. Yes.
Please, and next page.
Here we have the bacteria that is resistant to the antibiotic.
Is it the same colony?
No, It's a different one, which is resistant. This one is resistant to the antibiotic. Okay? Yes. So we made double-blind experiments. So we were given some bacteria. We didn't know if they are resistant or sensitive to a given antibiotic. We made our test. We give a table. Okay, this one is sensitive, the other one is resistant, and so on. And we had quite a high success rate, about 90% of success. In one case, the bacteria died during the transportation from the hospital up to the laboratory. In another case, it was quite special. But in any case, we see that it's relatively we can trust the method. So once we saw that it works for bacteria, of course, we tried with plenty of other living organisms. We used Candida albicans. Here you can see these Candida albicans that are deposited onto the cantilever. You can see here the cantilever. The small dots are the yeast on its surface. If they are alive, they induce oscillations, and once we inject a drug to which they are sensitive, the oscillations stop. If we deposit osteoblasts are cells that are building bones, okay? And so if the cell is attached here, you see the cell attached here at the very end of the cantilever. If it's alive, it shakes. But if it's dead, in this case, there are no oscillation left. We also made the very same experiment with nerve cells. And here again, if the cells are alive, they induce oscillations of the cantilever. As soon as they die, these oscillations are stopped. Then we went downwards. We tried it with proteins. And the protein we were used in this experiment is topoisomerase 2. So it is a protein that is taking a filament of DNA, it cuts it, opens it, takes another filament of DNA, passes through, and then stick the very first broken filament together. So this protein, to accomplish this, has to move a lot. Okay, they have plenty of conformational changes. And so we deposited this protein onto the cantilever. We add ATP that makes this, that is the fuel for this protein. And here is what we could notice. We have the protein onto the cantilever. If we increase the ATP concentration in the solution, we see that it moves more and more. And once we inject just buffer, the motion stops. Then if we add a drug that inhibits the conformational changes of topoisomerase 2, this drug is called aclarubicin. In this case, aclarubicin, you see, blocks the motion of this protein, and we can see it on the motion here. So we have a very simple way, actually, to see if a protein undergoes some conformational changes or not. And if we give also an ATP that cannot be hydrolyzed, in this case, nothing happens, and we have no oscillations onto the cantilever. So we have seen that we can detect plenty of different type of motion, and the question now is to know where is this motion coming from. So concerning the very first experiments, you remember I was telling you that it was done on E. coli. E. coli has this flagella. The flagella moves quite quickly and very probably induce shaking of the cantilever. And so we tried to increase the sugar concentration with this bacteria, and if we give them more sugar, they move more strongly, and we can see an increase in the motion of the cantilever, the oscillations of the cantilever. So concerning the mammalian cells, as you know, we have a cytoskeleton. The cytoskeleton is responsible for the motion of the cell and also gives the shape of the cell. There are different components. We have the actin filaments, intermediate filaments, and microtubules. So we tried drugs that inhibit these components of the cytoskeleton. We had the best results with cytochalasin, which depolymerizes actin filaments. So if we have an osteoblast onto the cantilever, you see it makes the cantilever oscillating. If we induce cytochalasin, after a while it will start to stop the motion, but it recovers slowly and then the motion comes back again. So we know that at least actin filaments play a role in these oscillations. So since we were very happy with these results, and we started to discuss with different hospitals to implement this detection method for bacterial resistance. And we noticed that there was a problem. You know, it's the price of the instrument. If you want to buy just an AFM to make just this type of measurement, it's very expensive because you don't use the scanner, you don't need the imaging software, and so on. So we decided to build our own prototypes. And the traditional AFM cost about 200,000 francs. For 5,000 francs or less, we are making a similar, this type of machines that make the same job. Here is this device. There is the laser, which is here. Then we have a mirror here. Underneath we have the liquid cell, which is the space in which we have the cantilever. And then there is inlet-outlet tubes, which permits to inject different solutions in there to see how the bacteria will react to those. And finally, we have the detectors, which are these two photodiodes you remember about which I was talking just before. And finally, some control electronics and a small camera. So now this instrument is going to be implemented in our university hospital in Lausanne, but also in La Plata in Argentina, where we have a collaboration with them. So this apparatus will be compared with the traditional techniques of antibiotic susceptibility tests, and we will see if it really helps in everyday life in a hospital. So to finish, just as you can see, we have a device that can rapidly detect bacterial sensitivity. Okay, it can be used for screening in hospitals. It can also be used for antibiotic research, but also we can imagine to deposit cancer cells onto the cantilever and inject different anti-cancerous drugs. In this case, we could have some kind of personalized medicine in oncology. Also, we could use it, we could imagine to use it for drug discovery, to have different proteins and to inject some chemicals and see if they induce conformational changes. But there is also one possibility to use this instrument, which I like a lot, is searching for extraterrestrial life. Okay, as you remember, I told you that we can actually detect bacteria, yeast, mammalian cell, all type of living organisms. So we could use this device to look for extraterrestrial life, to send it on a planet. Okay, and we have quite an efficient and simple way to detect if something is alive or not in a chemistry independent way. We don't need to know anything about the chemistry of this organism. We will see if it moves or not, you know. And so we made some preliminary experiments. So we took a soil sample, you know, we deposited it just onto the cantilever, and we were looking if it's shaking. And yes, it does. And as soon we inject some poison to kill everything, the movement stops. And we also took some samples of the river that is flowing close to our institute, and we made the cantilever very sticky. We immersed the cantilever into this water, and we inserted it into the microscope, and we could see that it's shaking. And once we inject glutaraldehyde, which is a poison that kills everything, we see the motion stopping. Okay, so that gave me the idea to maybe to propose the instrument to go onto a rover. Yes, it works. So we could imagine to have a system like this, where you take a sample of the terrain on Mars or another planet, and then you make a small filtration to select just the smallest particles. And yes, after the filtration, and if there are any living organisms in there, they should be green, of course, they would stick onto the cantilever. And if they are alive, in this case, the small motion that they have should induce a bit larger motions of the cantilever. And if we illuminate now this cantilever with the laser beam, we should see the deflections of the laser beam, as you can see it here. And if we have our two-segment photodiode that is located here, you see we illuminate different segments of the photodiode, and we can quite easily detect the motion. So I don't know if you had the opportunity to have a look at Jupiter, a small telescope. Yes. So you can see Jupiter has 4 moons, Io, Europe, Ganymede, and Callisto. And Europa is the second moon, and it's believed that it is covered by an ice crust, and underneath there is a liquid ocean. And if you look at the images that were taken by Galileo missions, in this case you can see some kind of icebergs, that look to float on this ice. And so NASA made a call to propose instruments that could detect life, because they are planning to send a robot onto Europa. And so we responded, and we were lucky we got the contract with the NASA. So we are now building the apparatus that could go onto Europa to detect life. So we didn't win for the moment. There are several other groups, but we are in the race, and we are very happy about it. And yes, I'm sorry. Yes.
This atomic force microscope is quite soft device, and what do you think? Can it withstand the space journey?
Yes.
Because the G load when the rocket lifts off is quite huge.
Yes. Yes. So, yeah, yes, there are already some AFM that were sent on Mars. It was working on Mars. And there was also another one which was sent on this comet, Philae. You remember that was there was a robot that landed onto a comet, and so there was also an AFM that worked properly. So it wouldn't be the very first one. And in addition, it's much simpler than the previous one, so we hope that we can make it. So. Pardon me?
So it will be survived, this approach.
Yes. So for the moment, they think that the mission will consist in just digging snow from the surface of Europa at 10 centimeters underneath the surface, because at this surface the radiation has no effect anymore. The big issue on Europa is the radiation because of Jupiter. And so we couldn't survive on the surface of Europa for more than a couple of minutes. But the material that is located about 10 centimeters underneath is almost not submitted to this radiation. So they will take it out, melt this ice, and then go into the different type of detectors that will be protected from radiation, and that would be at a temperature of maybe 5 or 10 degrees centigrade, because the ocean that is underneath is very probably at those temperatures. Yes. So as a conclusion, we have a device that can detect the susceptibility of bacteria to antibiotics in a couple of minutes, and also a very simple and light device that could be sent in the solar system to look for living organisms. And finally, the acknowledgements. Most of the experiments were done by Petar Stupar and Giovanni Longo. Wojtek is making the prototypes, he's building them. Alexander Kalauz and Ksenia Radutic gave us some samples and helped in calculating some results. And Jules Bergroeb and Giovanni Dittler are also the head of the different departments that collaborate in this project. And thank you for your attention. If you have any questions.